Our drug tenofovir is a nucleoside reverse transcriptase inhibitor that is prescribed to patients with a human immunodeficiency virus, better known as HIV. Question we propose is study the structure of tenofovir in comparison to deoxyadenosine and understand how tenofovir acts as a HIV-1 reverse transcriptase inhibitor via ligand mimicry. HIV is a retrovirus that brings in its own transcription machinery which includes integrase, protease, and reverse transcriptase. The enzyme we focused on is the reverse transcriptase, which contains two catalytic domains, the polymerase domain and the ribonuclease H domain. HIV infects cells by using the polymerase domain of reverse transcriptase to transcribe single-stranded viral RNA into a RNA-DNA complex. The ribonuclease H domain then breaks down the RNA, so the single-stranded DNA can return to the polymerase domain, where it will become a double-strand DNA. The viral DNA gets incorporated into the host DNA. And then this DNA gets transcribed into RNA. The viral RNA will be translated into viral proteins and packed itself into a protein capsid. The protein capsid will leave the current cell and infect other healthy cells with HIV. Incoming DNTP gets phosphorylated twice by two different kinases to become triphosphorylated. Reverse transcriptase recognized phosphorylated DNTP from our cells and added it into the viral DNA backbone. This forms a covalent bond.
This next picture shows our drug, tenofovir, working in a similar fashion as the DNTP. It gets phosphorylated twice by two different kinases to become activated. This allows tenofovir to be recognized by the reverse transcriptase. The reverse transcriptase gets fooled into adding tenofovir instead of DATP. Tenofovir is able to successfully get added by reverse transcriptase due to ligand mimicry. As you can see, the structures are very similar to each other. Tenofovir and DATP both contain the same adenine nitrogen base. When activated, both tenofovir and DATP have similar phosphorylations, with the only difference being a phosphonate group on tenofovir instead of a phosphate group. The major difference between tenofovir and DATP, and the reason our drug is successful, is the missing deoxyribose sugar group on tenofovir. This missing deoxyribose sugar group is replaced by a methyl group instead.
The following mechanism shows how DATP gets added to a growing viral DNA chain. The proton falls off in a proton transfer step due to the physiological basic pH of the environment. This creates a more nucleophilic oxygen that can attack the first phosphate group of the DATP, causing the last two phosphates to dissociate. This forms a covalent bond between the deoxyribose sugar group of the previous DNTP and the phosphate group of the DATP. The deoxyribose sugar group of this deoxyadenosine acts as a dock for the incoming DNTP group, and this process continues, forming a successful viral DNA strand. The mechanism of tenofovir's addition to the viral DNA backbone is very similar to that of DATP's addition. An initial proton transfer leads to a more nucleophilic oxygen on the deoxyribose group of the attached DNTP, again due to physiological basic pH. This oxygen then attacks the phosphonate group of tenofovir, causing the last two phosphate groups to also dissociate. This bond is a covalent bond. When an incoming DNTP tries to add on to the viral DNA chain, it is unable to find a site to bind to because tenofovir lacks the deoxyribose sugar group. Normally, DNTPs are rapidly getting added onto the DNA chain, but in this case, tenofovir blocks the addition by acting as a cap, causing termination of DNA elongation. The following shows the active site of the reverse transcriptase in the polymerase domain, which is made up of a magnesium, which is shown here, and the following amino acids: lysine 65 and 219, aspartic acid 110 and 185, arginine 72, and glutamine 151. Together, these amino acids interact with tenofovir to add it to the growing viral DNA chain.
Our drug, tenofovir, is successful in that it helps patients with HIV build up their immune system by slowing the progression of HIV-infected cells. As we described earlier, this is done by preventing the elongation of viral DNA. Typically, patients with HIV who take tenofovir show no negative side effects. However, a rare side effect that can occur is lactate acidosis, which is caused by mitochondrial toxicity. Tenofovir can be taken with other anti-HIV drugs as well. However, these drugs are not a cure for HIV. As of now, no cure for HIV has been found.